Hello, my name is Beatty. I'm an undergrad freshman biology student, and it's my protein project. So the protein that I was assigned were KRAs proteins, and my mutation is when glycine amino acid number 12 is replaced by aspartic acid or cysteine, so G12D or G12C. And this is what I have as the main protein. This is a KRAS quaternary structure protein, and the structure, of course, is quaternary, so it's not showing the primary, secondary, tertiary structures. But how I have subunits outlined in different colors, and especially the green is color coded to represent that this is the glycine amino acid number 12. So this is the regular non-mutated version of the protein. And then I have this indicating that this will be, this pipe cleaner right here indicates that this one is of course non-mutated, and that this is GDP, because GDP is the inactive state and GTP is the active state of the protein. So what what KRAS proteins are the relay proteins that are located inside the inner plasma membrane of the eukaryotic cells and neuron cells. And so that tells you where they can be found. They can be found on the inside of the plasma membrane of eukaryotic cells, so cells that have nucleuses, and then also neuron cells. The normal function of KRAS proteins is to provide instructions for making KRAS. So they get the information and they provide the instructions to build KRAS. They give the instructions to the nucleus. So they pass it from outside the cell inside the cell. And the protein is a GTPase that relays signals from outside the cell, like I said, inside the nucleus. And that way, since it's relaying from outside to inside the cell and giving signals, it's a relay molecule. So that's what I would categorize as a relay molecule. When this mutation occurs, when it's replaced by the aspartic acid or the cysteine, it becomes a lot more active, and it becomes in its active state permanently, because GDP is the inactive state and GTP is the active state of the protein. And while it's in its active state, it's constantly, like I already said, it's sending relay molecules inside the nucleus for them to construct the protein. It's causing uncontrolled growth, which we all know that uncontrolled growth in a cell, or the uncontrolled growth of cells, is cancer. So of course, this can cause cancer in the pancreas, liver, and many different cells that inhabit like quick growth and quick relay molecules. But when the protein is in its natural state, it looks like this. There's some signals, but it's not nearly as, you can see the difference between the GDP and the GTP. There's a big difference. There's a lot more relay molecules, or a lot more instructions being sent, a lot of signals being sent out compared to the regular. So the original, I constructed this out of styrofoam, and I use highlighters to illustrate some of the subunits based off of the diagram that I got offline. And then I used wire to illustrate GDP, and then I did the same thing in the mutated GTP version with the KRAS mutated structure. I decided to do the same concept, but the thing is that there's a key difference I have in both. One of them is that I wrote the glycine amino acid number 12, and then the other one I wrote the glycine 12D and the glycine 12C. So the original protein is polar, and the mutated protein is also polar, so there's no change in its polarity. They're both hydrophilic since they are polar molecules. The original protein is neutrally charged, and the mutated protein is negatively charged, so there is a difference between the charge. It goes from no charge being neutral to then, after there's modifications and there's the mutation, of course, it becomes negatively charged. The interactions for the original protein, so first and foremost, with any charge or polarity of an amino acid that you're going to have, there's going to be some type of van der Waals interactions. So that's one interaction that happens within this protein, because van der Waals interactions, of course, stabilize the interaction with weak bonds, allowing the amino acids to be close together without them crashing into each other and things of that nature. Other interactions that the original amino acid can go through are hydrophilic interactions, of course, because it's polar, and hydrophobic interactions also as well, since they tend to exclude water at times, even though it's polar. They can also be involved in ionic interactions, because KRAs contain several charged residues like glutamine that make it possible for the KRAs to bind a hydrolyzed GDP. because that's really one of the main things that it also does. It hydrolyzes, and that illustrates, of course, another interaction that it can do, because they're involved in hydrolysis, and KRA's activity mainly depends on hydrolysis. So that's, of course, those are a few interactions that the original protein can do. The mutated protein can do a few different interactions as well, because the original glycine G12 has a simple hydrogen atom as its side chain. It doesn't reform as many interactions beyond helping the protein stay flexible. But in comparison, this mutation adds a negatively charged carboxyl group, which is what makes this protein negatively charged, and this one is neutral. When the carboxyl group is added, it can form more ionic bonds with positive molecules because of that negative and positive attraction, and hydrogen bonds with water and other polar groups. And the G12C, so the second possibility, because it's either or, that mutation replaces glycine with cysteine, which has a thiol group, which is the negative SH group, and that can form disulfide bonds and be involved in redox reactions, making it much more reactive. These mutations, of course, change how KRAS works, locking it in an active state, which leads to the overgrowth. And while glycine side chain doesn't have any kind of reactivity, this is what usually happens when the mutation occurs. A few limitations that are happening with this model, I would say that it definitely was difficult to put together. I had an idea in my mind, it didn't necessarily come through as I thought it was going to. As you can see, there was a lot of mistakes that I did. I was trying to twine them up. I was trying to possibly do a DNA molecule to kind of illustrate specifically the detail of the gene and how it's changed and how it's a different gene being substituted in the mutation. But I just couldn't do it because it was really difficult. And then even this took me a while. And really the reason why I feel like it's a limitation is because the styrofoam was very hard to glue stuff onto. I actually had to carve out of the styrofoam. I had to carve out and then place glue inside, and then let the wires sit inside. Also, the wires are sharp, so the materials I used just weren't the best. I would say if I had to redo it again, I would use different materials that would be easier to stick, because it came apart a lot, and especially when it was time to bring it to class. The wind was blowing really hard, and these were falling off, and I had to put things back together. So structurally, it comes apart very easily. I would say that since it doesn't have an actual DNA molecule displaying the exact gene change, I showed the gene change with a different color here to represent the G12 and the G12C. And then here you can see it's green to represent the glycine amino acid number 12. So I color-coded it, but without me explaining that, you probably wouldn't know that. So that's another limitation I would say. I would say that's because it doesn't have that DNA molecule example, and that display is kind of hard to determine, and it's kind of hard for the naked eye just to see without explanation. And that leads me to my third limitation. I would say that just looking at this, it will be hard to explain what it is. Of course, I have a label on the back, but someone won't understand on first look. I would have to explain to them what it is, and I feel like that's a limitation to the model. Of course, with more time and maybe with more thought behind it, I will be able to come up with a better model. And if I have, of course, more materials, I'm very limited on materials in my dorm. With more materials, I'll be able to make a larger scale. But I purposely did go for a smaller scale because this amino acid is small compared to other amino acids. KRAS proteins are small compared to other amino acids. So that was one thing. Those are three limitations that I thought were limiting my my displays. Just to recap, it's a relay protein that is inside the plasma membrane, and it relays signals and instructions into the nucleus of how to make KRAS proteins. And the protein is a GTP paste that relays signals from the outside to the inside. The mutation occurs when glycine amino acid number 12 is replaced by aspartic acid or cysteine, G12D or G12C. And when this mutation occurs, of course, it leads to more activity, leads to uncontrolled growth, leads to cancer, leads to a lot of this growth and uncontrolled growth within the cell, and of course it spreads. So when the protein is in its original state, which will be the regular, no mutation, the GDP is inactive because GDP is the inactive state. And then when it's in its active state, that's what the GTP looks like. And regularly, these two switch. These two, the GDP and the GTP switch. So there's a control behind it because, of course, it needs to grow still. But when that mutation occurs, it never switches back. It stays in that GTP active state, and that leads to uncontrolled growth, unsupervised growth, which leads to a negatively charged atom, or not atom, molecule, which leads to this.